Welcome everybody. Thank you so much for joining us this evening at Highfield Hall and Gardens. So happy to see you all here in our lovely air-conditioned building. We are pleased to have John Henry and Izzy Gaw and May George here today to talk about curious critters at the Marine Biological Laboratory, or the MBL as we call it. They will talk about their cutting-edge robotics used to study and care for these intriguing aquatic creatures, which we'll have lots of time to look at after the presentation. From bizarre invertebrates to alien-like sea critters. Is it going to live up to that? Okay. Dr. Henry received his bachelor's degree at Lehigh University and earned his PhD at the University of Texas in Austin. After 2 postdoctoral positions at the University of Virginia and Indiana University, he joined the faculty of the University of Illinois as a professor in 1991. He spent many wonderful summers conducting research and teaching at MBL, and in 2021 he was lucky enough to move here to establish the MBL's Automated Aquatic Facility. He serves as senior aquarist and all-around ingenious designer. If there was an award for master tinkerer, he would surely have received it. In fact, he bought a house with an airplane hangar, not for a plane, but for his workshop of tools and toys to create, invent, and imagine. Izzy Gaw is the Automated Aquatic Facility Manager and has been at MBL for 1.5 years. And she was an undergrad at Skidmore College, majoring in biology and studio art. And she received her MS at the University of Hawaii and studied the deleterious effects of ingested microplastics on cryptogenic reef fish. Okay, everybody say that again 3 times fast. Okay. She was the director of the Natural Science Museum on Nantucket. And then luckily for us, she saw this position at the MBL, a combination of all her passions, and we have nabbed her now. She manages the facility, and she finds it a fun and fulfilling job, and she enjoys the research curiosity and her passion for scientific discovery. She considers her work to bridge creativity and science to address complex problems like how to culture new animal species. She and John are a formidable pair as they explore the weird, the wonderful, and the wildly unexpected world of marine science. Please help me join me in welcoming John and Izzy.
Thank you. Welcome everybody. Thanks for coming. I also want to introduce 2 additional folks who are part of the facility. May George is our aquarist. And Judy Venuti is in the audience, and she's a volunteer in the position at the So tonight what we want to do is introduce you to our relatively new automated aquatic facility at the Marine Biological Lab. I'll show you a little bit about that and some of the really fancy kinds of robotics that we have in play there to help us raise these really different organisms. And then I'll introduce you to some of them, and then you'll have time at the end to come and actually see them in person. And so let's start off by, of course, this is where we're located, the Marine Biological Lab. We are actually located off to the right here, so you can't see our building, but very close by. And so let's start by saying that a few years ago, so I came to MBL in 2021 to set up this automated aquatic facility, which is part of our new research organism initiative, which is basically designed to rear different kinds of marine organisms. In this case, they're all invertebrate species that are used in scientific research and in teaching. And to do that, we wanted to maintain large numbers of different species, large numbers of animals, and it's a lot of work. Animal husbandry is a lot of work. And so to make it easier, we wanted to employ various kinds of automation and robotics. And so these are some of the systems that we actually have in this facility now. We've renovated space in one of our teaching buildings, the alert building, and it is completely sealed facility, safe facility to house different kinds of species, including non-native species and even transgenic animals. And of course, we don't want those escaping to the external environment. So this is a secure facility, and it's climate controlled, and it does a very good job with its automated rack systems to maintain these animals year-round in continuous culture. So we designed these systems in partnership with a company here in Massachusetts called Automated Aquatics. And so they were designed and built in their facility to our specifications. And they have a lot of fancy automation to control water quality parameters, measure water quality parameters, and adjust them on the fly. And you'll see a little video of this facility in just a moment. So it's state-of-the-art. There's probably nothing like it anywhere else in the world. There are other facilities that employ automation, probably not to the degree to which we do. We had several goals in establishing this facility. We wanted, as I said, to maintain continuous cultures of these animals, so they're accessible year-round at any time. So these are self-propagating cultures of organisms, okay, invertebrate species, as I mentioned. To do this, we wanted to satisfy the goal of supporting our local researchers who come to visit MBL, as well as internationals throughout North America, and even in other parts of the world. And also our discovery courses that we deliver throughout the year, but mainly in the summer. We have a number of very high-level, interesting courses that teach different disciplines. So another goal was to create new research organisms, model organisms, if you will, inbred lines, established lines, and even transgenic animals that contain reporters, which allow us to study cell biology, molecular biology, developmental biology in live animals using fluorescent proteins that would mark specific cells, and you'll see pictures of that in a moment. As I mentioned, we can serve also in a capacity of a stock center to distribute these animals to other institutions. We can house other institutions' animals as backups in case something happens to their lines at their home institutions. They can come and get them and recover them from our facility so they don't lose them. And of course, we're always developing new robotic automation tools to support this work. And that's really a lot of enjoyment in that. So this is a little video which will take you down the hall to our facility. You'll hear some of the sounds of the water flowing and the pumps going and valves opening and closing. So all of these animals are raised in artificial seawater that we mix ourselves, store it, and it's automatically distributed to these rack systems. You can see the individual tanks that hold different species, different lines. You can see a lot of recording of data. Some animals are kept in the dark, others in the light. We have controlled light cycle in the room. And we'll zoom in and come back and you'll see a close-up of some of the tanks with the animals in them. You can sort of see how we have water being distributed to individual tanks with individual species inside of them. Okay?
What's the difference between transgenic and hybrid?
So hybrids would be, hybrid animals would be sort of genetically established lines through inbreeding, whereas transgenics would have other genes inserted, either deleted or inserted into their genome, which is what the term transgenesis is referring to. So there would be, for example, you could knock out a gene to study its function, or you could insert a reporter which would be expressed with a particular protein or under the promoter that controls the expression of that protein. So you could do it in a cell-specific, tissue-specific manner, and you'll see that in a second. Yeah. So these are the automated racks systems here. So they are self-contained units with recirculating water, artificial seawater. There's special filtration involved in this as well. There are heaters to regulate temperature as well as chillers. The salinity is monitored, pH is monitored. All of those conditions are monitored, flow rates are monitored and adjusted automatically. And this allows us to keep these animals, large numbers of different species, even in a single rack. And we can actually have up to 11 of these in that room, under very precisely controlled conditions. And it turns out that when you have very precisely regulated temperatures, salinities, that you get much better animal growth and reproduction. And so the health is much better in these animals. And this we've established very, very well compared to other forms of taking care of them. For example, if you had static tanks or aquaria where you're doing manual water changes and so on. And so we have accelerated growth and reproduction. This allows us to have continuous year-round cultures so we can have access to these animals and their embryos at any time. We're not really sort of subject to seasonal variation or seasonal reproductive periods. We have chosen species specifically for those advantages. We also offer, of course, remote monitoring. So these are all tied into the internet, so we can log in remotely. We can see all of the parameters. We can change them. We can adjust them on the fly. If there's any problems, we get alerts sent to us by email and in some cases by text messages. So we know immediately if there's a problem, and we can go in and see what's going on, and we can make changes if they need to, or if we know we have to come in. And sometimes, there might be a problem late at night, and if you can't adjust it remotely, then you come in at 2 a.m., for example. But these systems actually operate, they're very reliable, and it took us a while these are prototypes, it took us a while to establish these, but we've got all the bugs worked out of them for the most part, and so they're operating very, very well now. And so don't give us really too many problems. So again, it reduces labor, saves tremendously on the expense of maintaining large numbers of different species. They have special kinds of filtration, and you can kind of see some of those filter bowls down here at the bottom, and also located in this sump here where the water drains back before it's redistributed back to the tanks. These filters are designed specifically to prevent animals from moving through the loop so that they do not pass from one tank to another tank. And this is important when you have different lines of different transgenic strains and you want to keep them separated. Okay? Or if you have different species and you don't want them mixing between tanks. Okay? So it works very well. And so we can isolate and maintain, in this case, potentially up to 80 different species in one of these rack systems, because there are 80 of these 4.5 liter tanks. They can be configured in different ways so we can have different sizes of tanks. But these are the tanks that we've chosen to start with. So as you can tell, the animals we maintain are relatively small. Okay, you can keep them in large densities, so we can maintain a lot of them. In some cases, we have thousands of organisms of a single species in culture. Okay. Also, what we've done is we've developed our own automatic feeding robots. And so, again, saves us tremendous amount of work. So any kind of liquid food like phytoplankton, algae, for example, that we want to deliver, we can deliver it automatically around the clock on any schedule that we want. Even small live organisms like brine shrimp, for example, or rotifers or copepods can also be delivered automatically with these fluid handling robots. And so they pump them automatically to the system. They communicate back and forth with the main racks. So we build these ourselves. There are little microcontrollers in here. They're really fun to work with. They're not that difficult to program and get to operate. And we have little pumps, and we just pump, in this case it would be phytoplankton algae that pumps out of this Teflon line that goes into specific zones within those rack systems. Okay. And again, it mimics more of the real world conditions, environmental conditions. And so, again, just to promote the overall health and reproduction of these organisms in these systems. This is a sampling of some of the animals that we have in there, and we're going to focus on 6 of these species. We have everything from, in the upper left corner, shipworms, for example, which are, if you have a wooden boat or a wooden dock, are a substantial pest because they have symbiotic bacteria that allow them to digest, to eat the wood. We have little crustaceans like this little amphipod, Parhaela, which you'll see in a moment. We have snails like a little slipper snail, Propidula. I'll show you that. Different kinds of sea anemones and corals like Aptasia and Nematostella, and corals like Astrangia, which is our local hard coral, which we'll show you tonight, and other tropical corals we've also maintained. And we have some sponge species like this ball sponge, for example, and even sea squirts. Okay. So we'll focus on only 6 of these tonight, and we've brought all of them here so you can see them in person. So this is Haustinia. It's a little flatworm. It's not like your typical planarian flatworm that you probably have all learned about. This is a more ancestral organism. It's more basal. Like planaria, it's very good at regenerating. It actually has a population of stem cells inside it, which allow it to replace all of its body tissues. So it's a great system for studying stem cell biology and regenerative biology. And they're essentially immortal, so they're potentially a system also for studying aging. Okay? They reproduce sexually and asexually by fission, so they can just separate and split to form additional animals. They're pretty cool. They're called the three-banded panther worms. Here they are again. You can sort of see the banding pattern in some adults here. As I said, they have an internal population of totipotent stem cells, which can replace all of the tissues, all the somatic tissues, the body tissues in the animal. And this is a marker of stem cells called peewee, fluorescently labeling, so you can see where they're located inside the animal, which is outlined here. Another marker of reproducing, dividing cells. This is a gene involved in making DNA, and so required in those cells for undergoing division. If you cut these animals in half, they will regenerate the missing parts, and you'll see that in this next slide. You could, as long as you cut a piece that contains the stem cells, that piece will regenerate. Okay. If you cut a very anterior piece and there are no stem cells, it cannot regenerate. So here are some stages. This is the entire adult animal here. It's been cut on this red dashed line. So these are the anterior head pieces, regenerating over a period of days, as you see in the top. And these are the posterior tail pieces regenerating. So you can see the head piece will replace its tail, okay? And the posterior piece will regrow a new head. Okay? This is another organism we have called Nematostella. It's the scarlet sea anemone. You can see them feeding here. You'll watch them. They're eating brine shrimp. So you'll see them swallowing. They'll be coming down the gut in their stomach. They have little stinging cells that are located in the tentacles and in the body. And you'll see an example of that in a moment. You'll actually see them coming in contact and pull each other together, but of course they realize they're not food for each other, so they'll let go. Boing. These also are very good species for studying regeneration biology. They can regenerate parts. They have also a population of stem cells. They're referred to as eye cells or interstitial cells. And if you cut them, and you can see the experiment here, here's a Nematostella little sea anemone with its tentacles. Cut them through the body, and you can see over a very short period of just a small number of days, the posterior half will regrow the head. So you can see all the tentacles are present there. This is another sea anemone, which is Aptasia, which you'll see in a moment. It's also a very good species for studying regeneration. We do have some of those tonight too, but they will come up in a moment under a different context. This is actually an example of a transgenic Nematostella sea anemone, where they've inserted a version of the jellyfish green fluorescent protein under the control of a nematocyst or stinging cell specific promoter, okay, which is this nematogalactin here, galactin. And so each of these little fluorescent dots is one of their stinging nematocysts. And this is an alive organism. So these transgenic lines are very useful for studying the biology of this population of cells, for example, in this case, in a live animal. You don't have to fix them and stain them or section them to identify these cells. You can actually see them in live animals. Here's another animal we have. We've brought the local species. If you've been down to the beach, you probably have stepped on these and seen them. They're crepidulous snails. There are actually 3 different species of crepidulous snails that you can find here on Cape Cod. one of probably the most prevalent is Crepidula here that you see, which is the gregarious one, which forms these stacks. The adults actually adhere to each other, and they form large stacks. You'll see that here in this bowl in a few moments. They're very interesting because they are very good species for studying developmental biology, because you can get a lot of their embryos to study early development very easily. So if you pry them apart, we'll hope to demonstrate that, you can see all the egg sacs that have been laid by a female, which have been deposited on the individual below it, or the rock if it's attached to a rock. And you can pull those off and culture them, and you can do all kinds of experiments with these embryos, and they're a very, very nice system for doing that. But they're also interesting for other reasons. These snails are they're hermaphrodites. They actually change sex throughout their life. They start off, when they're small, they first develop into a male. And then as they get older and bigger, and you can see this is a male here, they become females. They're what we call the protandric hermaphrodites. So they go male to female throughout their life cycle. And there's some interesting biology to this, of course, understanding how this occurs, what controls it, how differentiation occurs, and so on. So they're a good model for that as well. And they're highly invasive, particularly that species, which have invaded other parts of the world, including the Mediterranean, the northern seas around France, Great Britain. Fortunately, they taste good. So they've started to harvest them. And so you can see this. Look at all of these snails. There's so many snails there. And they actually taste pretty good. So you can cook them, and you can collect them off the beach. Some people eat them raw. I would not recommend that. This is a Strangia. This is our local hard coral. There's actually a hard coral that lives in the waters around the Cape. It does not build reefs. It's not a reef-building coral, but it does build small little clumps. And we have some of those here. You can see their batteries of stinging cells in their tentacles here. These little dots that you see. It allows them to capture and immobilize their prey. And there's the mouth in the center. Of course, when they capture something, the tentacle will bring it into the mouth so they can swallow it. They're a very interesting species because, like many other hard, but not all other hard corals, they have symbiotic algae inside them. They're actually dinoflagellates, if you want to get to the technical term. They're called zooxanthellae. And they live inside the cells within these anemones, and those algae, of course, use the sunlight to produce energy, and they actually pass nutrients to their host corals, sugars and other things. And the corals, in some species, it's absolutely required. These symbiotes are required for survival. And this is a big problem for coral reefs around the world now. This particular species does not require them. You can actually photo bleach them if you put them in the dark. You can tamp down that population of those symbiotes. The coral will still survive as long as you feed them. We feed them brine shrimp, and they're just fine. But it's a good proxy model system for studying coral bleaching, the nature of symbiosis, which we really need if we're going to try to rescue our coral reefs. This is another. Let's see. This is Exaptasia, which is a brown sea anemone. And you see it. Maybe that was the same one that we just. No, this is a different one. You can't see the individual nematocysts here. They also have their symbiotic algae, but they don't require them. Okay. It's another model, but it's not a coral. It's a sea anemone, a related species. And these are the zooxanthellae, these symbiotic algae that live inside them. And of course, here's an example of, before a live reef and a reef which is undergoing coral bleaching, which is mainly dead. Okay, so if they cannot get the light, if the temperature is too high, or the pH rises, okay, because we're facing ocean acidification in various regions, they will lose their algae, and of course, without their algae, many of these corals cannot survive. Okay? So understanding this relationship, how it's maintained, what we can do to stabilize it is very important. This is a sand fleet parhalia. It's a little amphipod crustacean. These are interesting organisms. They have a, like us, they have a segmented body plan. They also have appendages. And so they're a very good model for studying segmentation and the growth of appendages. And you'll see, again, comparing humans, other animals like annelid worms, like earthworms are segmented. And of course, the crustaceans, amphipods also have segmented body plans. So they're a good system for studying the cell molecular developmental biology of the processes that result in segmentation and the growth of appendages. And these are transgenic parhalia, and Nippon is here in the audience tonight, the director of MBL, and of course these are animals that his lab generated. These have a fluorescent marker under the control of a muscle-specific promoter to drive the expression, in this case, of a green fluorescent protein in all the muscle cells. So you can see the muscles in the body and extending down into the limbs, okay, which control motion. And also another transgenic marker, which is I guess, I don't know if it's RFP or M-cherry, it's under the control of, again, I think this is specifically, is this in the muscle cells? Yeah, under a promoter to express a marker of a protein that binds DNA, so you can see all the cell nuclei in those muscle cells within the body. And again, this is in live animals, so you can study these processes in real time, live organisms, not fixed animals or sectioned animals. So it's these are very, very useful tools for cell and molecular biologists. And I think here you're just seeing, I think probably autofluorescence of the carapace. I don't know if that's what it is, but I imagine that's what it is. So you see a blue outline, so you can see the whole animal better. And so I'll just end with a thanks to the people who are in our team, who you've already been introduced to. We had one intern recently, Cyrus, who recently left us. He's gone on to other things. He's in college, and of course our donors who made a lot of this possible, and of course the generous support of the folks at MBL. And so I guess the next thing is to take questions, and then you can go and look at the animals in person. So thank you.